Well, good morning, everybody. Welcome to our second grand rounds of the academic season. And this week and next week are two very special grand rounds because they are in honor of two endowed professorships that have recently been developed. The one today is for Eric Hoffman, who has been named the A. James Clark Distinguished Professor of Molecular Genetics. And next week, it will be Mark Weissman, who has been named the Bernstein to the Bernstein Professorship in Community Health, and will be presented by Dr. Cora Bramble. To tell you a little bit about Dr. Hoffman, he did his PhD work at Johns Hopkins in genetics, and then did a postdoctoral fellowship at Harvard. And during that time, he co-discovered the gene for dystrophin, which is the underlying protein that is defective in Duchenne muscular dystrophy. And in the mid-1980s, this was the very first gene that was found to be associated with a human disease. And Eric has really made it his career goal to solve the problem of Duchenne muscular dystrophy and to cure it. And he has done and had many enormous successes, and is actually today going to be talking about the whole issue of orphan drug development and how to get a drug to market to benefit children with rare diseases in a more rapid fashion. This is something that may be important to many of you in the audience who are dealing with diseases in which there is not currently effective treatment, and in which you may have some ideas. And the idea of an MD working with a PhD has really been the way Eric has always worked. Although Eric is a PhD, his North Star has always been the focus on the child and how to improve that care. Lastly, I'd like to point to the importance of philanthropy and the importance of the philanthropy that has made endowed chairs possible here. When I came here in 1998, we had 3 endowed chairs. We now have 23 endowed chairs, 5 of them that are distinguished endowed chairs. And this has really allowed us to recruit and retain a number of superstars who are doing, some of whom are in the audience, who are doing spectacular research and clinical work. Eric really exemplifies this as someone who is a great scientist, who really has a focus on clinical care of children, and is a wonderful mentor whose trainees remain in his lab for decades to continue working with him. So let's welcome Eric Hoffman.
Thanks for coming this morning. I've sort of worked through a sort of historical story and put this in perspective. First of all, the Grand Rounds, as Mark said, tied to the Clark Charitable Foundation. I really want to thank them for their support over the last 20 years. That's the Clark building, all the construction, including this hospital. And in fact, this hospital launched Jim Clark's career in the public workspace. And now he's responsible, I think, for 25% of the hospitals being built in the country or something, a very large construction company. Typically, and most of these talks will focus on individual personal research, but Dr. Batshaw requested that I talk about spin-offs and Veralone. And why I bring this up is this is definitely not an individual personal research project. This really is a much broader As I put there, a disclaimer, Veralone was a community project of likely unprecedented scale. What I mean by that is not only community within Children's National Medical Center, but throughout the U.S. and throughout the world, and including both governments and foundations. So I'll try to paint this picture through the subsequent slides. Disclosures are really important when you talk about spin-offs. So Revirogen, which will be the topic, and Veralone, I'm co-founder, CEO, and president. It's a spin-off of Genetic Medicine, Children's National. First spin-off, I think, of Children's National. CNMC itself owns 35%, so there's a disclaimer there as well. Agata Biosciences was the second spin-off, and that is led by Kanaboi Nagaraju, but I'm a co-founder, and that's up in Halifax, Nova Scotia, doing mouse preclinical studies. And then Trins LLC is incorporated, but just getting going, and that's a spin-off of a clinical trials network we run out of Children's National. Okay, so to start this sort of weave this story, in 1985, which is when I started working in Duchenne muscular dystrophy, 86, DMD is really well recognized internationally. It's the most common genetic disease, monogenic disease. All world populations get Duchenne at the same frequency. For those of you who aren't, and I just realized talking to Mary this a second ago, I didn't put any introductory slides in Duchenne because I'm so used to speaking to audiences that are intimately familiar with it. But it's a muscle disease. Kids are born, it's 1 out of 5,000 live-born males worldwide. Animals get it just as frequently, so as soon as we can screen 5,000 pigs or dogs, we pick up Duchenne muscular dystrophy. The kids are generally born fairly normal. If you do actually like Bailey 3 scales, they'll have half the gross motor milestones, but parents won't really recognize that or physicians until they get into early school age. In early school age, like kindergarten, first grade, they're the last to go up the stairs and can never break into a run. And so they're often then referred to the parents saying, there's something wrong with Johnny, so take him to a physician, and then all you need to do is a blood test to see the CK, creatine kinase, which is thousands and thousands times normal. And so that just suggests a lot of a muscle disease. And that then leads to usually the diagnosis of Duchenne muscular dystrophy. The muscle then wastes progressively over the next decade or two, so kids are generally unable to maintain ambulation by about 10 or 11 years of age, and then die due to respiratory failure, usually before 20 unless mechanically ventilated. And so patients can live into their 30s now, but with very little ability to carry out any activities of daily living. So at this point, early on, there was basically nothing you can do. A parent was often, they'd bring in their kid to a physician, the diagnosis of Duchenne was made, and they were sent home saying, enjoy your time with your child because there's really nothing we can do. As Mark mentioned, in the late 80s, we identified the gene and the protein, and there was a lot of hype, and a lot of that got, a lot of hope, but a lot of that got pushed to hype by certain people and media, and maybe foundations as well, and there was a long haul then to try to turn this into therapeutics. Now, we did deliver things to patients and physicians and families along the way while we're looking for a therapeutic, and one is genetic testing. So with the identification of the gene, we were able to implement genetic testing into the neurology practice, and so neurology really became one of the leaders in terms of molecular diagnostics, and still is. And that became very accurate. We could do prenatal diagnosis. So there's a lot of hope, but little effort going into realizing this hope. No clinical trials, no pharma interest, no NIH interest. It's interesting that the funding of Duchenne was really split up between multiple NIH institutes with everybody not wanting to take responsibility for it because it's a childhood disease, NICHD, but it's a muscle disease that sort of fits into neurology and NINDS, but that's mostly neurologists. Neuromuscular is sort of poor second cousin, so it gets punted to rheumatologists over an NIAMS, and they're saying, well, we have to deal with arthritis, so let's forget about Duchenne. And basically just got stuck between. So in 2009, we moved from Pittsburgh to Children's National to take on the first version of the endowed chair. And then a key event happened, which just 6 months later, which is that son of Joel and Dana Wood was diagnosed with DMD here at Children's. And why that's important is because Joel and Dana were key, well-known lobbyists in Washington, D.C. So that initiated a key series of events that really completely changed the landscape. First, they set up a foundation. That foundation set up the first mouse drug trial center here at Children's. Really promising sort of what's viewed as the first personalized molecular drug development, which is oligonucleotides with skip-over mutation. That was on clinical hold at FDA. The company was doing a bad job. Through Children's and working with them in this group, we were able to get them off clinical hold, and that's now in multiple clinical trials worldwide. And we set up this clinical trial network called Synergy, which I'll come back to later. In 2001, that was just, I think, 6 months later again, they got 91 co-sponsors of the MD CARE Act, passes with 323 co-sponsors in the House of Representatives, something you don't see much anymore. And that was created the first NIH muscular dystrophy centers, first coordination of NIH research effort between all institutes, and the first CDC funding to look at incidence, work with state screening programs, et cetera. So then in 2003, we started getting DOD funding, so we got DOD appropriations. And early on, you don't get appropriations with your name, but we got children's named as appropriations of the Department of Defense bill, that we were able to run our own grant program together with foundations to find the most promising translational research, and that really got a lot of things going. And it's really created the seed money for this project I'll talk about in the rest of the presentation. So that's sort of, the plant is rooted, so we've created now in a short time, a couple of years, all this infrastructure for the first time to sort of now develop therapeutics. What do you deliver? What will a plant produce? And so the rest of this presentation will be one of those products, and there have been many, but I'll focus on what Mark wanted me to focus on. It's been called VVP15 based on how we screened for it. It's a horrible name, and it was recently just assigned a name by the USAN, a generic name of Verilone. So it'll be alternatively called those. Okay, so as I said, this was a lot of people involved in this, and really a very broad community project. So just a quick timeline in a couple slides. The idea was really that of John McCall. So he was previous Senior VP of Upjohn and Pfizer, and really probably the world's leading steroid medicinal chemist, and a really nice guy. His emails are hilarious, and says, just you know. And we had him as a consultant, as did NIH in many programs, and so he was on an advisory board. And he said, you know, I worked in this, you know that Duchenne's a problem of sort of the membrane cytoskeleton and instability of that. You know, we've worked on the steroid that seemed to stabilize plasma membranes. It was called this delta 911 background bone. It was brought to approval for both brain head trauma and spinal cord injury. Never really marketed that much, but he thought, let's bring back that chemistry and see if it stabilizes Duchenne membrane. Then the next key person is Kanabin Nagaraju, as many of you know. He's up in genetic medicine, and he had because he and I had both looked at fetal and neonatal Duchenne, and we found that NF-kappa B pathways, which is like chronic stimulation of innate immunity, were really cranked way up as soon as the kid was born, years before you see clinical symptoms. So the muscle was really struggling from missing dystrophin right from birth, and again, years before you saw clinical symptoms, 5 years. So we thought, well, let's go after this NF-kappa B. So if we can suppress innate immunity early on, will that help the muscle regenerate? Because it is a regenerative tissue, and we'll keep it from failing. And so he had set up the leading mouse model testing facility here, and he also had done an in vitro cell assay for NF-kappa B inhibition and started testing these compounds. And then Erica Rees, about 2008, had just gotten her PhD from GW with us in genetic medicine, and she said, I'll be your first employee. So she really, then when Revirogen spun off and we had identified this lead compound, she really carried the ball for quite a few years. And then he also was in the foundation space, the stakeholders space, this growing field of what's called venture philanthropy. And what most foundations, as you know, give out grants. And really a grant, typically, is you promise to do something, they give you money, and the money goes into a black box. And there's almost no accountability, no milestones, no deliverables, and it works to some extent. I mean, we all do that. But on the other hand, what if, you know, that's really the nonprofit, academic, ivory tower white coat that we're all wearing. On the other side is business, you know, money, you know, VCs, you know, everything for the dollar, where, you know, sure, you may be developing a drug, but really most importantly is how much money are you going to make off that drug. You know, maybe the patients and the issues and the stakeholders are somewhat secondary to money. What we're talking about here, and probably this program I'll describe is probably one of the best examples of venture philanthropy, is the space in between. So foundations will give us some of the biggest grants they've ever given anybody, but it's forgivable. So if everything falls apart, it just becomes a grant. But it's contracted as a 4-fold return on investment if it works out. So those foundations will get 4 times the money they put into it based on later drug sales milestones. So you're in this in-between space where it's about the cause and not about just the money. At the same time, there's deliverables, milestones. You don't get any of this money unless you achieve each milestone. And the first and a huge supporter of this program has been the Muscular Dystrophy Association. Jerry Lewis Telephone, first Sharon Hesterly, then Jane Larkindale. And they provided in 3 different milestone-driven venture philanthropy grants $3 million to this program. And it was the first grants like that that given out as an organization. Oh, and I should mention the CDMRP. As I mentioned, the Department of Defense was really provided the seed funding for this program. And then finally, our current rock star, which is Jesse Damsker, who's here in the audience, right there, with his guitar there, who really has been carrying the ball for the last couple of years at Revered and doing a wonderful job. And as I mentioned, a lot of stakeholders and a lot of groups funding this. We were one of the first 3 awardees of the NIH NCATS TREND program, Therapeutic for Rare and Neglected Disease. What this NIH program does is they cherry pick and look for the best orphan drug development programs, the most promising in the country, and they say, okay, we selected you. They selected us. What does it mean? Well, it means we give you no money, but we're going to, on our own dime, NIH dime, retest everything you say you've done. So they're going to validate every in vitro assay, every test, toxicity test, not everyone, but a lot of them, independently, and they'll pay for it. So basically, they're trying to de-risk your program by making sure every aspect of it is done well, robust, and validates. A lot of drug programs don't survive that process because they don't validate. But we survived well. We went through the whole process, and that then becomes a stamp of approval to foundations. Because foundations say, no, we don't have the expertise to evaluate this drug. We can bring in consultants and stuff, but wow, the government just de-risked it and showed that everything repeats. So then we have more faith in that along with our reviewers. And so another de-risking thing we did was Treat NMD. That's a European network. They bring together a bunch of consultants on both pharma, biotech, and academic drug programs. And they say, we're going to evaluate your program and say where there's holes and what you should do. Again, de-risking the program where those reports become available to foundations. And so then we had lots of stakeholder foundations. These are the foundations to date. I mean, I won't even name them all. FED is the local one. I already mentioned MDA. But throughout the world that then even collaborated to co-fund like the phase 1 trials. And then just this last month, just last couple of weeks, the FDA is giving us a $1 million grant for orphan drug development, NINDS $3 million for developing the phase 2 program, and the EU Horizons 2020, there were 400 applicants for 10 slots. And even though we're in the U.S., we incorporated in Britain, worked with our U.S. people, and so we got one of those 10 slots for $7 million to support the phase 2 program. So now I've gone over all this background. Just to summarize, $25 million in government and foundation support, a community-engaged and community-driven project, most peer-reviewed and de-risked drug program probably ever. I mean, how many people reviewed this program? I can't even count. Run by academic medicine. And this is important because being in the in-between space of academics, nonprofits, and for-profits, in this venture philanthropy space, we can take risks. We're much more risk tolerant. And furthermore, we'll do things for the benefit of all drug development programs. In other words, we'll try to help our competitors as a drug development company. If you're answering to stockholders, you can't do that. That's not your fiduciary duty. But by being in the venture philanthropy space, we're freed up to actually change what our mission is. It's not just money. It's the community and the patients and the stakeholders. So that pushes innovations, wishes to benefit all drug programs, and so we're operating in this space between public and private. Finally, just the last acknowledgments before I get into the story. At the end of the day, all this is driven by outstanding science. Really, Chris hires a wonderful postdoctoral fellow, worked with Kanamori Nagaraju and myself, who generated terrific. In the paper he published, I constantly hear from a lot of drug companies and scientists, it's one of the best, most robust drug development papers I've ever seen. Biomarkers become very important in this program, and in the 21st century cures that's being discussed downtown in Congress to push drug development, their number one thing is biomarkers. So I'll talk more about that in a second, why they're so important. But Yeshiv Hathout and Christy Brown have been wonderful there. Ed Connor really helped us as an early company with all his experience from MedImmune. on helping Children's National, and he was CEO for a few years. And then the clinical team. So we have the largest natural history study, 400 patients followed over 10 years. It's run out of Children's, but the PI of that is Craig McDonald at UC Davis. Paula Clemens is leading the 2A program. Kate Bushby Newcastle is running the 2B program. Avital Kanan here running the coordinating center, and Lauren Morgenroth is really doing a wonderful job coordinating all this within the Synergy group. Okay, so now that we've clearly established the broad international community involved, on with the story. So this is the story. Steroids. Prednisone, deflazacort is used in Duchenne in Europe, not so much in the US. Glucocorticoids is the general group. You learned about these when you went to school. Now, a quick history of this is 1948, first four patients with arthritis were treated with glucocorticoids, with adrenal extracts. And that was the fastest Nobel Prize ever awarded. The Nobel Committee have seen them use it as an example of how easy it is to win the Nobel Prize. With 4 patients, you can 2 years later get the Nobel Prize. I'm serious. So that was in 1950, and doctors Kendall, Reckstein, and Hench for their discoveries relating to the hormones of the adrenal cortex, their structure, and biological effects. Well, the Nobel Committee, you can look up the nice speeches, short speeches that were given by the Nobel Committee and awarding this Nobel Prize, and it was amazing how often they said, this is fantastic, it's magic, but you better do something about the side effects. I mean, the side effects were ominous. They were quite severe, and I'm sure many of you know of these side effects because many of you use glucocorticoids. Muscle wasting, muscle atrophy, adrenal suppression, all steroid hormones down. It screws up your aldosterone and salt balance. You get this cushingoid, bone fragility, immune suppression. And one thing you got to keep straight here in the rest of the story: anti-inflammatory is good. Immune suppression usually, not all instances, is bad. Some cases of glucocorticoids are used for immune suppression, but generally it's a side effect rather than the efficacy. Whereas anti-inflammatory is the efficacy. Okay, and that can be disorienting. So why we started working on glucocorticoids is because they are considered standard of care for Duchenne muscular dystrophy. So basically you gain about 2 to 3 years of ambulation if you start kids with daily high-dose glucocorticoids around 5 to 6 years of age, 7 years of age. Well, we just saw that muscle wasting, critical care myopathy. It's the greatest side effect coming out of critical care is muscle weakness, and that's tied to glucocorticoids. So you have a drug that we know causes muscle weakness that can be sometimes irreversible, and yet it's therapeutic for Duchenne. Never really made a lot of sense, but it also implies that maybe the efficacy of glucocorticoids is anti-inflammatory in Duchenne, but maybe that muscle atrophy and weakness is still there, and it's part of the side effect profile. And it just with that observation alone, if we can start separating out the efficacy from the safety issues of glucocorticoids, which really has been almost the holy grail. I mean, that's lots of pharma have tried. We got a recent review from one government panel, despite all these positive reviews, that said, we're not sure if we believe this because pharma should have done it before. And the grant was rejected. But everybody else says, wow, the data seems to be there. So that's what I'm going to talk about. Glucocorticoids are complicated. There are many different activities. Remember, they're just pharmacological glucocorticoids are just minor derivatives of cortisol. And cortisol is your diurnal hormone. So everybody this morning, a couple hours ago, about 4 hours ago, your cortisol's peaked. And they're starting to head down pretty good now, and by lunchtime, they'll trout. And that sets your wake-night cycle. It's also a stress hormone. If you get stressed, you shoot your cortisol up. And that's all glucocorticoids are. So really, it creates this conundrum. Why is cortisol, which goes all the way through plants, is throughout the animal kingdom, setting up these daily circadian rhythms of wake and sleep. And even plants have glucocorticoids that do the same thing with opening to sunlight and closing. Why are pharmacological doses a magic anti-inflammatory? And nobody's quite understood that. They're involved in immune modulation, hormonal regulation of all steroids. You need to understand these levels, peel away the levels, and then separate, and that'll be the theme of the rest of the talk, safety versus efficacy. So what our sort of hypothesis is there's a lot of good layers of the onion, which are efficacy, a bunch of bad layers that are safety. If we can start separating these out and fine-tuning the drug, get rid of the safety issues, then enhance the efficacy issues. So when we started this, there were two well-known sort of layers, or what I'll call sub-activities of glucocorticoids. And again, glucocorticoids do lots of things as steroids do, so I'm going to really separate out what shall end up being 4 or 5 different sub-activities. The one you learned in med school or as an undergraduate is called transactivation. That's what everybody learns about every steroid hormone. Steroid hormone is produced somewhere, it circulates through your body. Because it's lipophilic, it goes through cell membranes sort of generically. There's no receptor for it in cell membranes. It binds its cytoplasmic receptor, so only the cells making the glucocorticoid receptor then see the ligand. That ligand receptor then dimerizes, and that goes into the nucleus, and you influence gene expression. So there's lots of genes with promoter elements that have glucocorticoid responsive elements that will bind that dimerized ligand receptor complex. Same for testosterone, estrogen, all your steroid hormones work under that same general principle. But what happened is this increasingly through the literature seemed to be safety signals. So in other words, what you learned throughout medical school about how glucocorticoids work is a safety problem. It doesn't seem to be this efficacy with glucocorticoids. Instead, what became increasingly apparent is efficacy, the anti-inflammatory efficacy, is associated with a subactivity called transrepression. This is called transactivation because you're activating genes. This is called transrepression because you're repressing other receptor complexes from activating their genes, and it's mostly NF-kappa B. So remember that activity I said is present, screaming from birth. It's really the canonical innate immunity pathway. As soon as your body gets upset about anything and stimulates innate immunity, NF-kappa B complexes are just really being turned on, and that is a key pro-inflammatory pathway. So what glucocorticoids and the receptor do is the glucocorticoid still diffuses and still binds its receptor, but then that receptor interacts with NF-kappa B receptors and inhibits them. So NF-kappa B can no longer go in and be pro-inflammatory. So it's inhibiting the pro-inflammatory activity of NF-kappa B. And this seems to shut down the danger signals at their source, and this is increasingly recognized as a good layer of efficacy signals. So the brief history here was, as I mentioned, John was on an external advisory board. He said, well, this is a fragile membrane problem in Duchenne. This is a membrane stabilizer. Raju extended that because we didn't have good assays for membrane stabilization at the time. Jody Jaiswal's done those subsequently upstairs. But Raju says, well, we already know NF-kappa B is in there. We already know steroids work through NF-kappa B, glucocorticoids. So I have this cell-based assay. Let's see if these delta 911s still do NF-kappa B. We had recruited him from Hopkins, and he'd done this reporter construct, and so that worked, and I'll show you that data in a second. So here's that data. Basically, here's a cell-based assay looking at NF-kappa B reporter activity, and as you increase the concentration of VVP15, which became our lead compound, you see it tracks exactly with prednisone. So prednisone is a very potent NF-kappa B inhibitor. That's probably most of its efficacy as far as anti-inflammatory effect, and we're retaining that. Now we did, and here's Jody Jaiswal's work, did look at membrane stability. Remember, as steroids, because they're lipophilic, as they go through the plasma membrane, they spend some time there. And because they're lipid-like before they go into the cytoplasm. Well, what do they do to the plasma membrane when they're in the plasma membrane? So that's what this assay is. And what you see is prednisone, which is here. We're looking at how easily a cell is injured and allows dye into the cell. And this is the controls. And here you see prednisone exacerbates injury. So prednisone, that's actually a side effect, we think, and particularly for Duchenne, of prednisone, is it makes membranes even less stable. Whereas John's hypothesis was right. Here you see a dose-dependent stability of VVP15. So we've now changed the activity of this steroid, so while it's still anti-inflammatory, it's now stabilizing plasma membranes instead of destabilizing them. So here's just another, then we looked at the hormonal toxicity or this transactivation. Our goal, as I said, that's what you learned in medical school, at transactivation. We want to lose it because it's a part of the side effects. Here's prednisone turning on these gene promoter elements. Here's our drug DVP-15. So we have gotten rid of transactivation. How have we gotten rid of transactivation? I don't know if I have a structure here shown or not. It all had to do with one double bond, and what it seems is that when the ligand binds the receptor, our ligand, it won't let it dimerize. So we don't create the dimers. Now the single ligand receptor still carries out transrepression. It still inhibits NF-kappa B. But you need that dimerization to go into the nucleus and work on gene promoters. And so we seem to, by changing the chemistry of the steroids, have lost that ability, and that's shown here. And here we see, for example, pituitary cells, you want to lose things like adrenal suppression. So this is a marker for adrenal suppression, and here you see the prednisone and dexamethasone induce adrenal suppression with this marker, whereas here we've opened up that window considerably. So this was our lead compound. We screened about 25 drugs and came up with this. We may have a steroid-like drug with layers optimized. Let's see how fast we can run it through the paces. Let's see if we can do it as a public-private partnership through this venture philanthropy model. So one of the things that became very important in moving this quickly, and this is what I sort of mentioned in the learning objectives and the title, is this program became sort of a way to a proof of principle addressing this growing problem in medicine. The growing problem in medicine is drug development is getting really more and more personalized. And orphan disease is sort of used as an example of that. The problem is it's often quoted that it takes about $500 million in 15 years from start to bring a drug to market. Now, as Mark points out, in orphan disease, it's probably less than that, 100 million or something, but it's a lot of money and a lot of time. Well, in rare disease, orphan disease, you have very few patients to amortize those costs back. So that's where you see, like the last CF drug, it works like magic, but it's $400,000 per patient per year. And that's what they're, or 300,000, it's somewhere in between there, I think, depending on how much you weigh. And you'll see that on a lot of these orphan drug developments. Well, there's a conundrum here, because if we're heading towards personalized medicine, where breast cancer is not one type of breast cancer, but 10, 20, 30, where drugs are more and more personalized to specific molecular pathways, cancer is leading that field, but we're seeing it everywhere. Well, all drugs, almost by definition, become more and more personalized. Well, if you're getting more and more personalized, there's less and less people. Is our system going to afford $400,000 a year per patient for each drug we prescribe? It's not. So that's where both the U.S. and Europe are really seeing this growing problem, trying to figure out how to circumvent it. There's only one way to circumvent it: make it faster. So by moving the approvals much earlier in the process, you move much of the quality of life studies to post-marketing. It dramatically reduces the cost. And that's what they're doing now in both Europe and the U.S., these so-called accelerated approvals. But you need to build a robust case. And how do you build a robust case that you're going to, in the future, influence quality of life without necessarily showing it first? And that's where I'll show you how you do that. Biomarkers become really important in that. So this is the infrastructure we had put together. I mentioned the lobby efforts, this clinical trial network, preclinical testing facility. So the first thing we did was to see if this drug made the mouse model of muscular dystrophy happy. Okay, so that was done by Chris Heyer and Kanawin Nagaraju, and many people collaborating on that. So one of the side effects of prednisone, particularly for you in pediatrics, is growth stunting. Currently, when you go to a Duchenne muscular dystrophy conference, what most people say, you no longer see Duchenne, you see steroids. You see kids that are half the size of their peers, cushienoid, weak bones, you see steroids. And in fact, we see more and more patients losing ambulation because of the steroids, because they'll break a bone even though they're still ambulatory, and it's hard to get them back walking if you've lost that much muscle. So you see this growth stunting if you compare it to vehicle in prednisone, and BBP-15 does not cause that stunting. So basically, we've lost that subactivity or safety signal of growth suppression. Here's adrenal suppression. Would like to lose that, and indeed we do. Seems to we need to do more in vivo tests. Muscle strength. Now, interesting here, we see a dose. Here's muscular dystrophy mice, so they're weaker than normal. And we see this dose-dependent increase in strength with this drug that works. Now, prednisone, it looks like it makes them weaker, which it does. Now, this is absolute strength. But remember, we have very small mice, right? So if you normalize for weight and make this kilograms force per kilogram of mouse, prednisone goes up to equal BBP15. So prednisone causes small, strong mice. And that's probably what it's doing to Duchenne kids as well. Small, strong Duchenne kids. But what this says is if we separate out safety and efficacy signals, we'll end up with larger, stronger Duchenne kids, hopefully. This is immune suppression. As I mentioned, we want to keep anti-inflammatory but lose immune suppression. And so here you see the prednisone induces immune suppression by multiple assays. And these doses go up. This is 6 times prednisone dose here, and still no immune suppression. Prednisone in mice causes some major heart problems. You don't see that as much in humans, but here you see the fibrosis induced by prednisone, and a BBP15 doesn't do that at even higher doses. As I mentioned early on, muscle wasting is a bad side effect. Critical care myopathy is one of the most obvious examples of that. And here you see prednisone induces these molecular signatures of muscle wasting, and BBP15 does not, like Jesse Damsker's and Blythe Billingham's work. Now, another sort of surprise, so let me just summarize. Transactivation, what you learned in medical school, we wanted to lose it and we seem to have lost it. And that goes with a lot of side effects, so we seem to have lost those. Transrepression, we want to keep that. That's the anti-inflammatory activity, and we seem to have kept that. Membrane stability, we seem to have improved, right? Because we've taken prednisone, which is a membrane destabilizer, turned it into a membrane stabilizer, and that's good for Duchenne muscular dystrophy. Now, this fourth activity was sort of a surprise, and that had to do with pleuronone. Pleuronone's an MR antagonist, okay, mineralocorticoid receptor. Mineralocorticoid receptor, as you know, controls, normal ligand is aldosterone coming out of the adrenals, and that controls your salt balance, among other things, okay, through your kidneys. Well, a pleuronone as an antagonist is used by many people, you know, particularly cardiologists. And there was a nice recent paper that says the pleuronone really helps improve heart function in Duchenne muscular dystrophy. So that's great. Well, prednisone is an agonist of the mineralocorticoid receptor. A pleuronone is an antagonist. So what we did is early on a series of in vitro assays to look at different nuclear hormone receptors. Here you see prednisone as an agonist of the mineralocorticoid receptor. That's probably adding to its safety signal. That's a safety problem. So being an agonist is a safety problem. An antagonist becomes an efficacy feature. And that's what we've done. BBP15, while being an agonist like prednisone of the glucocorticoid receptor, has turned into an antagonist of the MR. In fact, we just got data from Raju's lab and Agata showing, in fact, it looks like this BBP15 fixes the heart problem in mouse models of muscular dystrophy. So we've really turned this drug and taken a safety signal and added a pleuronone, it's almost the same potency as a pleuronone, on top of it. So basically, to summarize this, retention of transrepression, gain of membrane stability, loss of transactivation, and MR antagonist instead of agonist, and all this should combine into sort of a customized drug. Well, focus on Duchenne muscular dystrophy. What about all these other indications for glucocorticoids? So we've tested quite a few: asthma, multiple sclerosis, inflammatory bowel disease, Lori Conklin's group, and also with I'm blanking on the head of surgery. Sandler's. He's right there wherever he is. There he is. What's your name? That happens to everybody, doesn't it? And arthritis as well. And again, Jesse's done a lot of work in Raju's lab. So it seems to show efficacy, at least mouse models of all those. So lots of tests to do, costs a lot, need a bunch of people, takes a venture capitalist, but again, we've used the community approach. I've mentioned the NCATS program, Treat NMD, lots of grant reviewers. Last month was 11 million, we're up to 25 million. The FDA and EMA have been extremely helpful. I mean, you can see it in their eyes. You go to them as a bunch of academics, and they're saying, wow, finally, it's not about money. And we're getting so much positive feedback from them, and lots of informal communications that nobody else seems to be able to get. Here's a list of all the funding that's gone into this, all the different foundations, all the different grants. So I'll just briefly in the last few slides go over the clinical program. We're about done with the phase 1 clinical trials, so we've started low, 0.1. Prednisone dose is about 0.7 mg per kg per day. So we're going all the way up to 20 mg per kg. We did in the SAD, no side effects, and we've done the fed cohort. The MAD, we're going, which is, this is single ascending dose, this is multiple ascending dose, so this is 2-week daily treatment. We've just finished the 3, and we soon start the 8 mg, and that 8, again, is 10 times prednisone dose. For the 2A and 2B, which is what we've just gotten funding for from both Europe and the US, we're focusing on innovation, data-driven clinical endpoints, because we have these huge natural history studies we've done for the last 10 years, data-driven pharmacodynamic biomarkers really done by Yetri Pathout, great papers coming out of his group. And we've done parallel FDA EMA guidance, which again isn't often done, but we want to see how we can push that parallel guidance. We've hired, are collaborating with international key opinion leaders to run the program. Kate Bushby and Paula Clemens are going to be the directors of the clinical trials. Last couple of slides, this is SAD data, so PK data, pharmacokinetics. You see low dose to high dose in normal volunteers, beautiful dose proportionality, and little variation between subjects. Half-life of 2 hours, similar to PRED, which is another thing to point out. When you give daily PRED, it's gone in 2 hours, or half-life of 2 hours. It's gone in 4 hours. So basically, most drugs you want to sustain sort of a therapeutic level, and you dose to keep a certain level. Prednisone, you don't do that. You've never done that. In fact, if you try to do that, you increase side effects rather than efficacy. So really, it's this pulse of glucocorticoids that gives you the anti-inflammatory efficacy. Nobody's ever really explained that. Now, we have a lot of projects working on that, and I think we're getting there, but again, we look just like glucocorticoids. I mentioned this cooperative international research group, 24 recruitment sites in 12 countries that we run with Avital Kanan here at Children's. This really was kept alive by DoD appropriations. Any clinical trial network, as Roger and many know, is feeding the beast. How to keep it going and in order to keep the infrastructure intact. And we've enrolled over 1,000 subjects to date. And then, as I mentioned, this biomarkers program becomes extremely important in the clinical trial, and I'll just explain that in the last couple of slides. This is the key thing. Well, I'll skip over that. Biomarkers, and that's the last sort of conceptual thing. Why is the 20th Congress obsessed with biomarkers? I mean, if you want to read, the only piece of legislation I've really found fun to read is the 21st Century Cures legislation. Look it up on the web. It's being debated downtown. It's how are we going to sort of transform drug development to make it faster and cheaper? And it's really well written, really interesting. There's, what, 4 pages of Bayesian statistics. Bayesian statistics, you guys, that's, you know, additive probabilities. It's like going to Las Vegas. It's basically saying, you know, if we take the probability of this event and the probability of this event and we do additive probabilities, what's the cumulative probability? Well, why is that relevant to drug development? Well, because if you think about it, your clinical trials are all based on your primary outcome measure. You have to pick one thing. In case of Duchenne, are they going to walk longer over 6 minutes? And call that, you know, what you're going to judge your drug program on. Well, there's lots of cases we've all seen where there seem to be hints or maybe good evidence that drug might be working, but it failed the primary outcome measure and drug development is stopped. Is that really fair? Isn't that sort of betting a whole drug program and betting with patients? Why not take a series of outcome measures and have a combined probability? Has to be compelling, has to make sense. That's Bayesian statistics, and the government wants that brought into clinical trials. So why biomarkers are so important is really just paint this picture. What we're talking about is pharmacodynamic biomarkers mostly. And what that means is we give a kid, say, VBP15, and within maybe a couple weeks, maybe 2 months, we can see in the drop of blood the signature of what that drug is doing, both in terms of efficacy and safety. And we know and can prove that that biomarker signal anticipates later clinical benefit and later clinical safety based on clinical outcomes. Okay? So, you know, just to give you one example that you're probably familiar with, if you give prednisone, you create insulin resistance. Insulin resistance is a side effect. It's not good. So we see, and Yetri found in looking at 200 patients in the one, 100 somewhere in that, Duchenne patients through a natural history study just published in PNAS, that you treat with prednisone and actually insulin goes up compared to other kids because you've induced insulin resistance. Well, isn't that then a molecular signature of insulin resistance? It is. Well, you can do that for adrenal suppression, insulin-resistant, cushingoid, aldosterone, all these different safety signals, and you can do it for efficacy signals as well. What's the pro-inflammatory pathways? How is the drug suppressing those? And those signals you see in the blood acutely and objectively. And objectively is important and acutely is important because a patient really by placebo effect can't change their blood. That's much harder. Well, it's not completely true. Anti-inflammatory, you can, with placebo effect, get some decrease in pro-inflammatory proteins, but generally not. And so that's where this program, we're really pushing that. Our primary outcome for safety is BMI change versus previous steroid trial. We ran out of here. Efficacy, our primary outcome is time to stand. And so here's our current regulatory path. We've triggered both parallel EMA FDA guidance. We're currently ending our phase 1 by the end of this year. We're starting the phase 2A actually in January next year. And this 2A and 2A extension, the 2A is just a 2-week treatment, MAD, multiple ascending dose, to look at PK and safety and tolerability. But then because they're children, you need to enroll them into an extension study because you have to have intent to treat. So you need, and FDA said, you have to have an extension study, which means 6 months on drug. So that's here. So we're having kids for about 6 months on drug. Well, we're well-powered to see differences in BMI and well-powered to see differences in time to stand. But this has no control group. We can't use a control group in a PK study for Duchenne in a child. So you have placebo effect. So even though we're well-powered for our clinical outcomes, how do we rule out placebo effect? Any guesses? Biomarkers, right? We have the blood test and we have the whole panel for both safety and efficacy that Yettrieb's defined. They should rule out placebo effect. And so we think we can go to the FDA and EMA at the end of this 24-patient extension study and say, look, isn't this story compelling? We see changes in safety. We see the efficacy retained or even better. Our blood biomarkers rule out placebo effect, give us an accelerated approval at that point, and we have already kicked off the 2B, which has a placebo arm and a steroid arm, prednisone arm, throughout Europe. So this becomes a confirmatory trial in the post-marketing phase. So that's sort of how these accelerated approvals are working. So this is the last slide, acknowledgments made possible by the community. I mentioned these as I went, and all the foundations participating in that. And again, thank Genetic Medicine and the Synergy Group and all the people upstairs and throughout the country working together on this. Thank you very much.
Thank you, Eric. That was a really wonderful
talk and very exciting. And there are a couple of points I'd like you all to just take away from this before hopefully folks will come up to ask questions. We all think of NIH funding as the gold standard, and in one sense it is. But as Eric has said, there can be a whole community that can help you with your research, ranging from the NIH to private foundations to individual donors to venture philanthropy, all of which Eric has used over the last decade to really move this project forward. And frequently it can be getting that NIH or similar grant that can, as he said, serve as kind of the gold standard where the philanthropists will then give you money. The other point is that it does take a community, and we have a number of infrastructures here that really support you in your research, and those include the Clinical and Translational Science Award, which is directed by Lisa Gay Wolford here, and has a number of the cores that Eric's group has used, the biostatistics core, the cellular imaging core, the genomics proteomics core, study design core. All of those things can help you. And in addition, there are a large number of pilot grants that are available in the $30,000 to $50,000 range that can get you started. So when you have an idea, don't let it die in loneliness. First of all, get other people to work with you on it. come and visit us in CRI to see how we can help you move things forward. And if you're interested in the issue of drug development, we have a world's expert here in this. So not only think about what Eric said, but how it could apply to what you're doing yourself. Now let's open it up for questions. I'm sure there are a lot of interesting issues that would be brought up. Maybe we'll start with Roger. Roger, coming up, you deal with this issue of rare diseases in terms of brain cancer and development of new drugs. And can you kind of reflect on what Eric has been saying? And
well, I think that every foundation we work with now wants to get into this area of philanthropy and venture capitalists. And it's been a very interesting movement for most of the foundations that I've seen that want to make this investment. And they keep putting out, and maybe you want to talk about that, the CF Foundation as being the paradigm for someone who worked with both industry and with academia to move their program forward. Did you use the CF as any kind of a modeling system of how to move things faster through the process?
Well, the CF Foundation, as you point out, has really been a leader there. And a lot of that, how we've used that is models of their contracts. So what is a venture philanthropy contract look like? And in fact, many foundations, CF foundations have done all that due diligence, come up with contracts that seem to work, and those are then replicated in other foundations. So in effect, almost all the contracts we've signed, which is going upwards of 10 or so, for venture philanthropy are modeled after the CF Foundation.
And the only other comment I'd make is the one interesting thing is you've taken some of the competition between drug companies out of the equation. We presently are at an issue with, for gliomas in children, that there's a set of drugs that hit the RAS-MAP kinase pathway, the MEK inhibitors, that are extremely active drugs. And we are stuck in a situation, we have 4 drug companies with 4 different drugs, none of which will communicate with one another of where to push them forward. Each of them having their own other drugs that they may combine with those drugs, but not allowing them to combine with other drugs that other drug companies come. So this is a nice model that you could sort of hopefully avoid that. But that's my big frustration, is to get industry partners not interested in my problem, but to stop competing with one another.
Yeah, the orphan drug space is just exploding. I mean, it's really, if I would have asked you 3 or 4 years ago if there'd be competing drug companies in pediatric brain tumors, you would have probably laughed.
We would have been happy to have that.
Right. So yeah, I think with exon skipping, which I mentioned in passing, which is another drug development program we're very actively involved with, I think the key thing is to facilitate everybody's program as an academic as best as you can. And I'm often called by the community apolitical. So it's really hard to make me, if you're viewed as political, then things start falling apart, particularly as an academic. So you really have to play all fields equally. We have some questions from the WebEx. So how do you address global toxicity in humans, liver, other organs? I mean, toxicity is a huge aspect, both preclinical and clinical in any drug development program, and FDA is particularly concerned about toxicity. So this whole series of tests, I mean, I didn't go over them, but I have some slides of the number of both preclinical and clinical tests you have to run, and they're truly intimidating. So you really make sure you fulfill all the criteria that FDA and EMA has for what are relatively standard, both preclinical, meaning animal, and clinical human toxicity tests. And that's why we're doing the phase 1 clinical trials in normal volunteers. Adult volunteers is predominantly for toxicity. The second question is, how do you know systemic distribution? Well, steroids generally distribute quite freely because they're small molecules and lipophilic, and they cross the blood-brain barrier, et cetera. So these show a wide biodistribution. I think you have to do formal studies both in animals and in humans to show biodistribution, but that shouldn't be an issue with BBP-15 and hasn't to date.
Eric, congratulations. This is a wonderful tour de force of drug development. I have a couple of questions. One question is, there's so many anti-inflammatories in the market today. What is unique about steroids versus other anti-inflammatories that made it work so well?
Well, steroids, glucocorticoids, have been around for, what, 70 years now. And, they still, how many of you use glucocorticoids all the time in many different disorders? And, almost everything's off-label because I've never really often run trials, and they're cheap as dirt, which is another thing to consider. But, I think if particularly in children, if you take something like Duchenne or inflammatory bowel disease, where the side effects may overwhelm, in some instances, the efficacy. And it's always a concern. Lori, when she prescribes glucocorticoids, I think, is concerned about that because of the side effect profiles that often come with that. And yet they're still used because there hasn't been a good replacement. So it's possible, we certainly haven't proven this yet, but this could be a safer and possibly more effective because we can push dose because we don't have side effects. It doesn't seem to be alternative to glucocorticoids. Now, it's going to have to justify an increase in cost, obviously, but that's another thing to point out. In working in this venture philanthropy model, if it ends up costing $400,000 a year, you're going to find me dead at the hands of parents in Washington, D.C. in an alley, right? Because we developed with this with the community. If the community can't afford it, right, we're going to take a huge hit with that. It's almost disingenuous, is that the word, that you're doing that. So even with the possible pharma partners, we discussed right at the first meeting, we can't make this an expensive drug. We don't want this to be an expensive drug. And to some extent, we're fortunate in benchmarking against prednisone because prednisone is cheap as dirt. That's who we're competing against.
Eric, one last short question. Could you make a comment about this is an hypothesis-driven drug development? The idea was that that's already known, maybe it worked. What prevented you or others to do a wide chemical screen? Is you don't have the right platform to do thousands of chemicals for Duchenne?
Yeah, so that's a good point. So I think this, if I got the question right, I should mention that the patent protection around VBP is not pristine. What we have is a patent picket fence around methods of use. We don't have composition of matter. The reason we don't have composition, even though it's a first-in-human drug, the reason we don't have composition of matter is because our co-founder, the steroid chemist, John McCall, back when he worked for Upjohn in the 70s and 80s, mentioned this, what ended up being our lead, as an intermediate in another synthesis. So just because it was mentioned in a patent as a structure, you can't get composition of matter. So then, we are at a somewhat weak position, but we have a fairly good, what's called patent picket fence, but it is a concern. So why don't we do what Mendel just said, is screen another 1,000 drugs to get composition of matter on one. And we have planned that. We've made some compounds. But in this venture philanthropy model, we don't have the financial wherewithal to actually go after that. And actually, one of the only grants that hasn't been funded in this program is trying to do that screen. So really, we hope with a business partnership that can get some money in the bank for somebody to sell the drug later, a established pharma, that will allow us to do exactly what you said and then have composition of matter as well as possibly better drugs.
Yes, Eric, this is fascinating. Of course, like most people in this room, I would like to know when I can start prescribing. But do you know, or are you working on the effect on other inflammatory diseases, asthma, CF, any neutrophilic or eosinophilic inflammation? What do we know about it?
Yeah, one thing that was nice with this Clark that made this distinguished endowed chair is they're also providing $700,000 a year in pilot money for orphan drug development that you're all welcome to apply for. So that's 4 years, $700,000 a year. So one of the pilots was a CF pig trial of VBP-15, the first CF pig drug trial of any type, and that's funded by the Clark Group. So we're pursuing CF. Zena Quezado is doing mouse preclinical studies in sickle cell, and IBD we think is probably the next indication we'll actually pursue, but there's lots of indications. We have orphan drug applications written up for like 3 other disorders that we just haven't submitted. We really need a hint of efficacy, at least through biomarkers first.
It's 9 o'clock, so I'd like to invite any of you who'd like to talk further with Eric to come up, and thank you very much for joining us. It was perfect.